It is with great pleasure that I'm going to introduce Paul Turner. Paul received his BA from the University of Rochester and his PhD with Richard Lenski, who's known for his long-term bacterial evolution. Online, outside of his many honors and professional achievements, I've seen Paul referred to as an all-around awesome dude, friendly and generous with his time, and I'm putting my money on his ideas. Paul is the Elihu Professor of Ecology and Evolutionary Biology at Yale, and also a microbiology faculty member at the Yale School of Medicine. Paul has provided a service to our communities in many ways, including mentoring over 60 undergraduate students over the years and many doctoral students. He has donated his time for conversations with non-scientists through mass media, including several pieces in The Scientist and public lectures such as this. He has also given a series of online lectures for iBiology to convey viral biology to those who can't always make lecture series like this one. If you want to see more of him, I encourage you to YouTube those. Paul has demonstrated leadership on issues of basic research, as well as for public and health policy. For example, he was an appointed member on the Committee of Gene-Derived Research in Non-Human Organisms at the National Research Council. He's also donated his time as an appointed member of the Biological Sciences Advisory Committee of the National Science Foundation, and he served on the council of the American Genetics Association. And my favorite kind of fact is that he was also a member of the US delegation at the joint USA-Russia workshop on infectious disease. He encourages us to do research not just in our lab, but out in the broader society. Paul's research, though, is wide-spanning, and it's hard to summarize it in just a couple minutes, but generally he's interested in viral evolution and ecology. We know that viruses from Paul's work are important for parasites and human evolution. But today I'd really like to focus on a specific thread on some of his recent publications on the renewed potential of phage therapy. So phage therapy actually is about 100 and almost 50 years old at this point. And it ran out of favor right after World War II when he discovered antibiotics. When it was used, it was thought to be maybe effective, but the problem is that it's not broad-reaching. As you know, now that we have antibiotics, we think the world's great. But we also now realize that we're hitting a cliff where our antibiotics are starting to not work. And we have to face the reality that we're entering into a post-antibiotic regime. In other words, in 50 years, the antibiotics that we use right now to solve common infections like strep throat might not work. Can you imagine going back to a time like that? So Paul has some great ideas about how we can possibly combat this. And to use the idea of phage, can we use phage themselves to force bacteria to still remain susceptible to the antibiotics that we use them? He raises the possibility that we can actually face this post-antibiotics era by harnessing the natural power of its own predator, the phage. Paul today will tell us about the good, the bad, and the ugly of the phages and viruses.
Well, thanks everybody for being here on this must be a slow night in Cambridge for all these people in the room. I'm honored to be here, delighted to hold forth for the next 45 minutes or so and kind of share with you a lot of my own passion and love for the virus world. So in a museum setting like this, which is just an awesome and an inspiring place to catalog and display Earth's biodiversity, I'm into the kind of biodiversity that you can't see with the naked eye. So that's a lot of what I want to focus on tonight, is that there's this amazing biodiversity that exists on the planet. In fact, it is so vast, it's hard to even accurately measure how many viruses there are. So to me, it's kind of an endless playground to study the virus world. And as my wonderful introducer reminded, I am going to be able to talk somewhat about how viruses might save your life one day. So I'm trying to have you leave the room on an upbeat note, rather than think about all the pain and destruction that viruses can exact on the human population. Okay, so with no further ado, thank you very much for being here, and let's get started. I tend to pace a lot when I talk. I hope that's okay. I'm told as long as I don't go into this corner and hide out from the cameras, we'll be all right. So I have given outreach talks frequently, and I often like to use this theme of one of my favorite movies. And I decided to give a similar lecture in a friend's non-majors course at Yale. And maybe it was a size about this, class size about this one. And I asked all the students to raise their hand if they'd ever heard of this movie or seen it. And I'll tell you, like 3 hands went up. So I want to know, how many people in the room? Okay, that's not bad. If you're a young person, you have to talk to an older person next to you to keep up, that's okay. See, they're running from the room because they've never seen the movie. So I do love this movie. I like Clint Eastwood westerns and other westerns, and I'm going to eventually get to this theme. But first I want to start with this, and this is going to echo something that I mentioned a moment ago. I was fortunate to travel to Alaska recently with my family, and we saw many of these charismatic megafauna, right? These are very recognizable creatures for the most part: moose, caribou, bear, and these are the so-called Big Five. So if you're on the equivalent of the safari in Alaska, you'd like to get a glimpse of each of these organisms, which is not easy, especially the sheep, because they live up very high in the mountains. So that's the kind of visible biodiversity that people walk through the halls of this museum and they recognize some creatures like these. Not so much the ones on the right, which far outnumber the visible biodiversity on the planet, but this is some of the invisible biodiversity. So this very strange looking creature, that's Giardia, and that's the reminder that if you go camping and you want to drink water that has not been boiled, you may not come back from that camping trip, unfortunately. So these are cartoons. Obviously, if you can make out the one at the bottom, that's a bacteriophage. I'm going to talk a lot about bacteriophages, but most likely in the last third of the talk. This is an HIV particle. So most people have certainly heard of HIV. If you haven't heard of bacteriophages, you'll hear a lot about them tonight. I think this is influenza virus particles. And so this one requires some explanation. There are some microbes that interact with other organisms, and they basically will take over the behaviors of those organisms and make them do something else. So this happens a lot with ants, and it depends on what the microbe is, but in this case, this is a fungus that has infected this ant. It's altered its behavior in some ways I won't get into, but eventually this fruiting body will come out of the head of the ant, and that's going to spread spores, and it's going to go and infect more ants. Pretty gruesome. But that kind of stuff fascinates me, so I hope you're ready. So what we know is that we live in a microbial-filled world. That's kind of what I'm emphasizing. I'm going to give you an outline of my talk, but I'm going to give a few slides first to talk about microbes in general before I focus on viruses. So clearly in my lifetime, and this is very recent stuff that has come about, but now it is in all biology textbooks. The fact that in and on your body, there's a microbial diversity that we're still in the process of characterizing it. It's so vast and interesting, and person to person, you've got differences. People live in different geographic regions. They have differences in their microbiomes, right? So specific to her body, there are different bacteria, fungi, and viruses living on and in the human body. So the real compelling thing is how much are these microbes driving the bus? So there are these really compelling studies for which the results are exciting, but we're not quite sure what it means. If you take someone who is obese and someone who's lean, and you take the microbes from their body, you can put them in a mouse, and the mice will take on the traits of the human. And then you can take the microbes out of the mice, and you swap them into the other mouse, and they become either obese or lean. So this has everybody thinking, oh, well, I'll just take a pill eventually, and I can get rid of that very expensive gym membership. I'm looking forward to that, but sorry, that's not happening anytime soon. This kind of stuff it gives us hope, but it also gives you pause to think about, are the microbes in my gut telling me to eat when they are hungry, even though I'm not? That's kind of where this research is going. But there's all kinds of other things I could spend all night talking about. How much your microbial fauna and flora, whatever you would like to call it, that's associated with your body, how much of it is actually setting you on a pace for things that are pretty dire in the future, like higher risk of heart disease and cancer. So open questions. Here's one that's gotten a lot of press, and that is most of us in this room, but it depends where you grew up as a child, a lot of humans live in pretty clean environments. And there's a popular idea now that we're actually living in too clean of an environment on average, such that in childhood especially, we don't see worm infections. Whereas people who live in very rural environments, and entire countries are full of people who live almost entirely in these rural environments, they see worm infections at a young age. And it primes your immune system properly, so your immune system doesn't turn on you and cause autoimmune disease, right? So this could be at the root of many mysterious allergy rates in places like the USA, whereas in places in the developing world, this is not occurring. However, it's very complex in terms of this might be happening, but there's also evidence that the time at which you introduce solid food to babies is also something that dictates how much of a risk of allergy they will have in the future. So this is a really compelling idea, might have to do with exposure to microbes at a young age, and yet a reminder that we live in a very complex environmental world, and all kinds of things can influence your future health, especially risk of allergy. So this is what I want to cover tonight, though, these 4 bullet points. Not to anger, annoy anybody, but I'm going to assume that there's people in the room who don't know what a virus is. So thank you for being here tonight. I guarantee after you leave, you're going to know what a virus is. So I like trivia contests. My wife thinks I'm sometimes pretty trivial, but the point is I like to go out with friends, and we often lose, full disclosure. It's not like we're really good at trivia contests, but I like to build them into my talks. Why is that? Because it's going to make you pay attention, and it's also going to make you challenge yourself. And it's not like I'm going to test you on things that I've said already. Instead, I'm going to challenge you for 6 questions to see whether you're going to get them right. And sorry, there are no door prizes. It's only bragging rights. So basically, keep track of those 6 questions along the way. Turn to your neighbor and boast that you got all 6 correct, and maybe they got none of them correct. And we'll see who leaves the room proud that they did well on this quiz that's built in. And then I'm going to cover viruses in a way that the movie takes the title of the movie and flips it on its head, right? The characters are introduced in the opposite order as the title of the movie. So I'll talk about ugly viruses first, bad ones, and then good ones. Again, so you leave the room on an upbeat tone. Could a virus save your life one day? I'll spend some time on that. So to begin, the upper portion of this slide and the lower portion of the slide are fundamentally different. So there are 3 domains of life, and they're all cellular-based. The bacteria and the archaea have essentially a one-cell body plan, but they have some differences from one another that I won't get into. On the right is an example of you. It doesn't look like you, but it is a relative of yours. It's another eukaryote. It happens to be a typical cell taken from a eukaryote, which these can be single-celled organisms as well as also multicellular organisms like us. So the big difference is that eukaryotes have a nucleus in their cell, if you remember that from your biology course. So viruses are most easily defined by what they're not. They don't have cells. They have a body plan, but it doesn't have anything to do with a cellular body plan. Instead, they are generally described as proteins that are protecting nucleic acid. And the nucleic acid could be DNA or RNA, some variant of those 2. So essentially here on the left, we've got a bacteriophage of a particular virus family, and you can see it's really cool, I think a really beautiful structure to it, a morphology. But I'm pointing to this capsid. This is a protein shell that protects the DNA from degradation from environmental factors. And on the right might be something that's more familiar to you, that's an influenza virus particle. It happens to have also protein that's shielding nucleic acid, but in that case it's RNA. So the simplest lay version of what a virus does is shown in this diagram. So if there are any virologists in the audience, I apologize by reducing it down to this level, but this is going to have everybody essentially understand what I'm talking about. A virus particle encounters some cell, and if it can bind and the genetic material gets in, essentially that cell gets taken over as a little factory to make more viruses. That's all you have to know. So that could be happening in a bacterium. So if this is a bacterial cell, this virus, I'll talk about this more, can infect this bacterium and eventually kill it and release more viruses. Or it could be happening in your respiratory tract or your liver. If there are viruses that are specific to certain parts of your body, certain organs, this is happening. And it's going to cause symptoms, usually. A lot of virus illnesses are symptomatic, not asymptomatic, but not all of them. This is just a general idea of how viruses replicate. So despite the fact that I can kind of give you a common basis for how they replicate, I think that there's a beautiful amount of morphology in the virus world, and that's what's shown here in this cartoon. So maybe you've heard of some of these. Herpes viruses are quite common and successful in infecting humans. Rhabdovirus, that's rabies virus is a rhabdovirus. Again, this is one of those, what I think are quite beautiful bacteriophages. And then others have very humble morphology, like this tube. That's not very interesting, is it? But that's what that virus has. So notice one thing here. This is a scale. This is 1 micron, this bar. That is 1/1,000,000th of a meter. So I can now impress upon you that most viruses are submicroscopic. You can't see them with light microscopy, and that's why they were missing from the detection of microbiologists for a very, very long time. I don't have a slide of it today, but I should, in case you've heard of it, there are some viruses that are quite giant-sized. They are literally equivalent to the size of the smallest cells, and that was something that was recently found. So we can talk about more about those in the Q&A if you want. First question. So we live in a very virus-filled world, but I haven't told you where viruses come from. I'm an evolutionary biologist. I care about origins of organisms. And here are some ideas for where viruses could have come from. Did they appear billions of years ago, preceding cellular life? And then they evolved and remain to the current day? Are they parasites that came out of cellular genomes? So this is a DNA replication picture. So imagine some little bit of this goes rogue, and it has an autonomous state next, and it just goes around infecting other cells and replicating. Are they devolved cellular life? So you take them all the way down the path of cells evolving, and then they reduce all the way down to viruses. Again, for the youngsters in the audience, this is Devo, de-evolution. Do they come from outer space? So this is literally the title of a paper. I know some scientists are in the audience because I recognize you. I haven't pointed you out yet, but I might. I know that none of you wrote this paper. This went through peer review. It says, is bacteriophage 5X174 DNA a message from an extraterrestrial intelligence? I will hold my comments. And the morphology of a bacteriophage relative to a lunar lander, maybe that paper has a point. Or is the answer E, that more than one of the above is probably true? So I'll give you entirely 10 more seconds to think about this. And I want you to answer this. Keep track of how you're doing, because at the end you're going to have a score sheet to tell you how well you did. So I would have you shout out the answer, except it might embarrass you when I give you the true answer. What do you think it is? E. So basically, more than one of these is probably true, but not D. Okay? So I would say that virologists that care about this question are pretty split on what the answer would be. And as I was mentioning I was saying this to someone before the talk, the problem is viruses leave absolutely no fossil record. It's very hard to go back in time. And even genomes, if you know anything about genome sequencing, it's possible to create phylogenetic trees of relatedness between things that you've sequenced. But the signal for viruses is very, very weak, especially if they're RNA viruses that mutate a lot. Okay, so I hope, despite what you shouted out, I hope you got that right. Viruses grow very quickly. So this is a Drosophila life cycle. Drosophila were one of the earliest organisms to really be used in evolutionary genetics and genetics research in the early 1900s because they grow very quickly. They're convenient to grow them in the lab. A lot of amphibians, these are also eukaryotes. They grow very, very quickly. That's a handful of amphibian eggs of some species that I don't know. I'm a virologist. I don't know anything about amphibians. Bacteria also impressed me with the rate at which they grow. They were the first microbes that I studied as a PhD student. And each bacterial cell is female by definition. It can split into two and create two daughter cells. very efficient if there's enough food to go around to feed them. Now this is a bacterial cell for which one of these phages has infected, taken over the machinery of the cell, and this cell is about to burst open and release these particles, even though there are a bunch of other ones that naively believe that they could get in. This is obviously a cartoon, but this reminds me to say in your respiratory tract or somewhere in your body, each one of the cells in that organ could be themselves a virus production factory producing thousands of particles. And no wonder you would have a sore throat, right? Now, they thrive in all environments that you would find life, which is pretty impressive, given that in the extreme cold, the extreme heat, the deep ocean, under high pressure, in all these environments you find viruses. And they tend to outnumber any cellular life that you find there. So, ergo, or therefore, this is not a very controversial statement. They're the most numerous thing on the planet. The human global population size is pretty impressive, and worryingly so. But viruses at any one time, it's estimated that 10 to the 31 particles exist on this planet. I'm not a mathematician, but this is a bigger number than this one. I just want you to understand that. So, factoid number 2 and question number 2. If you remember from basic biology or genetics, if you've had it, is that the DNA in your cells, of which you have plenty of DNA in your cells, it's in a compacted form. It is possible to unwind it. So if you took all the viruses on the planet and you unwound their genetic material and you laid them end to end, how far off of the surface of the planet would this extend? Would it go to the moon, to Mars, to Pluto, which I still respect as a planet because I learned it when I was a kid, the Perseus cluster, or to infinity? I'm hearing A, D, the Perseus cluster, 250 million light years. So if you are an amateur astronomer, go look up the Perseus cluster and the fact that this huge cloud is moving around the Perseus cluster, and it takes millions of years to go around the whole thing. But I digress. The point is that that was 250 million light years off the surface of the planet, which is very impressive to me. Now getting to the meat of the talk. So brace yourselves, because some viruses are quite ugly, okay? So Tuco is he's the first main character who's really introduced, and they have this long list of pretty bad things that he did as he's about to be executed. But don't worry, kids, it gets better. What is very evident is that humans have been suffering from virus illnesses for a very long time. And you can find this as long ago in hieroglyphics and other remains of, say, ancient Egypt, where this man has a very characteristic malformed leg and foot. Okay, so poliovirus exists in soil. It's very easy to encounter it. The problem is when it gets in your body, well, actually, it can get in your body and go through your gastrointestinal system and you never know it. But what it'll do inside your body is invade your nervous tissue. And if it does that as you're growing and developing, it can lead to malformed limbs. And of course, it could be deadly as well. So in the remains of some mummies, you'll find also club feet and other characteristic malformed limbs of poliovirus infections. Now, this is a little harder to see, I know, but the mummified remains of Ramses V, this pharaoh, he had smallpox, and he died from smallpox. That's what we know now. I'm sure they were mystified then. So this is a more recent photo of someone with characteristic smallpox lesions, but these are quite evident even though you can't quite make them out in his mummified remains. So that shows us that at least as long ago as ancient Egypt, you've got, in the time of the pharaohs, you've got smallpox and poliovirus being pretty common. And I won't go into more examples, just know that this is something that humans have been dealing with for a long time. And then you can think about how impactful has this been on humans for mortality, and this is some of the ugly statistics. So it's a little hard to really rank the 4 examples that I'm going to give you, but some diseases are so-called endemic. They're in a region, and whatever the host organism is, they're suffering from these diseases. Viruses can cause epidemics, where they'll come in and cause a lot of destruction and mortality in a host system, and this has happened in humans. So a particularly deadly form of influenza virus in 1918 called the Spanish flu killed some 50 to 100 million victims. Impressively at a time when commercial flight was not happening. And this virus managed to sweep the globe. It's that infectious and that contagious that very, very many millions of people died. So the next one I'm color coding in black rather than in red. All the ones that are truly viral are going to be in red. The Great Plague in Europe in the middle 1300s killed maybe 28 million people, or some 40% of the European population at that time. So that's a bacterial illness caused by Yersinia pestis. But many people believe that you couldn't get that much death due to that bacterium alone. It probably was that people were living under very adverse conditions, and they were therefore susceptible to lots of other things that might infect them, including some viruses. So this is a little sketchy in terms of how much of this was due to virus versus bacterial illness. The New World smallpox epidemic, circa 1520, it's hard to know how destructive this was because we didn't have good census data on Native American populations at that time. But clearly we know that almost entire tribes were wiped out due to this when Europeans came in and they brought smallpox to the New World, and there was no Native American resistance to it. And then the last one is what I tell students at Yale and elsewhere, is that this is an epidemic that we are currently living through. So the AIDS epidemic started around 1981, at least that's when it was defined to have started. 77 million people perhaps have been infected since then, 35 million people have died, and currently 37 million are living with HIV. So pretty impressively gruesome numbers. In some of that is a topic that I like to study in my own laboratory, and is a very difficult thing for biomedicine to predict, and that is what is going to be the next thing that sweeps into humans and causes a lot of destruction. So that's a very difficult thing to predict, as you might imagine. Some of what we try to examine is the ability for viruses to jump between host species. And some viruses are quite good at jumping directly into humans, unfortunately, and causing deadly diseases. I've got examples here. So the tricky thing with emergence is it really happens three different ways, but it's thrown together into one catch-all term. So you have something like these flying foxes, these bats. They're really bats. They are daytime foragers, and in places like Australia, they are nesting or roosting in the trees and also foraging in the trees, and they will pee and defecate, and it'll fall to the ground. And this could be happening on a farm where pigs are rooting around, eating, and they take in the virus. And it may not kill, in other words, they'll take in a virus. So, I forgot to say, bats are quite good at carrying viruses, and in this case, Nipah virus can be taken up by the pigs, and humans who are farming the pigs, or if you eat undercooked pork, you can get a Nipah virus infection, which can be deadly. So that's a form of emergence. It's going from the reservoir host ultimately to humans, and that's why we care about it. So HIV, I kind of hinted at this before, HIV is a very interesting virus. It's actually evolved many times independently. So SIV, there are many SIV strains that are in chimps and other primates like pseudomangabe monkeys. And as long ago as 1930s, these viruses were getting into people, and yet the chains of transmission were pretty weak. And it took all the way until the 1980s before we realized that this virus was in people, and it has a very long latent period, like 10 years, before you'd know you're even infected. And then when that happens, your immune system starts to crash because the virus is destroying your immune system cells. So the point is it's very good at going from non-human primates into humans, and it happened independently several times to evolve into HIV-1 and HIV-2. So that's a form of emergence. And then on the right, even viruses that we see all the time and that circulate in humans, like influenza virus, you can get much more deadly strains, like I told you happened in 1918. The real fear is that the main reservoir of influenza viruses on this planet is in waterfowl, especially. There's much, much more of that virus in birds than there is in humans. And humans don't do very well when they get avian flu. It can be deadly. So that's a looming high likelihood of possibility that you'll get a deadly flu outbreak happening eventually in humans, and the source will probably be from birds. So I can give you many examples to kind of sober you before I try and give you some good news. Zika virus is yet another virus that has been known for some period of time in Africa. More recently, you've got the spread due to global warming of mosquitoes that are capable of transmitting this virus, and therefore the virus is spreading to places that it hadn't been traditionally, and more humans are encountering it. And you probably know that one of the very unfortunate outcomes is that this can lead to microcephaly in children. So you don't have the brain case developing properly, and therefore brain size is limited. So that's where we started. Let's just take a breath. It's going to be okay. Now we're going to talk about the bad viruses. Here's where I introduce Angel Eyes, or Lee Van Cleef from the movie. Again, go watch the movie if you don't know what I'm talking about. So this is a quote that my mom would say around the house. They can put a man on the moon, but they can't cure the common cold. So I was born in 1966. It was that precious time when we were trying to get to the moon. We did it successfully, and yet I think people were kind of awestruck that we could do that, and yet there were some things that were quite annoying in medicine that we didn't solve. And I'm pretty certain my mom didn't come up with this quote, but she heard it somewhere and she would just say it around the house. Rhinoviruses and other viruses, rhinoviruses, by the way, have nothing to do with rhinos. Rhino means nose, right? So some viruses make you sick, but they won't kill you. I won't go into gender differences and how much people whine when they get a cold. I whine a lot when I get a cold. I really sometimes wish I was dead, but it's not, actually these can be very dangerous to people who have asthma. So there are people who have a very debilitated state from having a common cold. And yet, you're ideally staying at home, you're not going to school, you get through it over the course of 3 or so days, but some colds, as you know, can last weeks. So it's very annoying, and it is not something that has been attempted to be fixed through vaccines lately. There was actually an attempt at it some time ago, and it didn't sell. That's because the viruses evolve so quickly that you produce a vaccine, and basically it's ineffective because the viruses have moved on genetically, and they're not targeted by the vaccine. So other viruses that are not so important in this country but kill people around the world, I'm going to give you an example of this, rotavirus. Now this is a virus that if you get infected with it, you do wish you were dead. You basically have inability to keep fluids in your body. If you can, I won't get more graphic than that. So this, I know this intimately because my daughter, when she was 2 years old, we were living in New Haven, she had a rotavirus infection, and she had to be on an IV for rehydration therapy for at least 24 hours. I think it might have been 2 days. And it gave me a clear understanding of a lot of people live in this world without access to clean water. And all you really need, usually to get through this infection, is rehydration with clean water. But lots of kids don't have that. So this is perhaps 5% of children around the world. This is the thing that kills them, is rotavirus. It's untreatable by drugs. Vaccines exist. There's been some challenges getting those vaccines really to the people who need them. So think about, a lot of viruses are pretty bad. We don't struggle with them so much in this country, but they're pretty bad, and a lot of harm comes, including death in other countries. Now I want to move on to talking about other organisms than humans. I like to impress people with this because it makes them think that I read literature, which is not really true because I'm a sci-fi buff. But I did read Watership Down, and it's a pretty cool fantasy novel. And in this book, and if you've ever seen the cartoon version of it, they talk about white blindness as something that if the rabbits in the colony, or whatever the group of rabbits are called, has this, then they kick them out. So this is a real thing. I don't know if you can make it out. This rabbit has an unfortunate kind of tumorous growth on its face. That's because it's infected with this virus that causes myxomatosis, and eventually the rabbit's going to go blind. Okay, so this is a virus infection. So myxoma virus was purposefully released in a country because they had a rabbit invasion problem. Okay, what is it? Yes, Australia, which is famous for its biocontrol strategies that don't always work. In 1950, myxoma virus was released in Australia. So unfortunately, when I blow this picture up, it's so low resolution that you can't see what it is. But it is a watering hole surrounded by rabbits. and you look in the distance and all you see is rabbits. So this is the state of Australia at that time. And they grew the virus, some scientists grew the virus in the lab. It was quite deadly in the laboratory, and then they released it. And it worked initially, and then they didn't quite factor in through basic epidemiology that a rabbit has to be infected for long enough and live long enough so that the chain of transmission goes to the next rabbit. And it sort of pushed the system back to a more moderate form of the virus. It evolved in the wild to be less virulent than you found in the laboratory. And basically the biocontrol effort failed. Okay, so now let's talk about the good. We're gonna get upbeat. I'm gonna tell you how viruses might save your life one day. And Clint Eastwood, who's supposed to be the good person, the good character, I forgot to say this at the beginning. Basically, all these characters are very, very flawed. That's why this is a classic movie. It has an almost nonsensical title because it categorizes the characters into these bins, and you see them moving from bin to bin, okay? Well, we'll just say that Clint was the good guy because he likes to be the good guy, usually in his movies. So we live in an area that's, well, I live in an area that's very close to the epicenter of Lyme disease. I know you're worried about it here as well. But it's an example of if you take a system and you throw it out of balance because some species are missing, you can create problems. So lots of deer live in Connecticut and other areas of the Northeast where Lyme disease is prevalent. And deer are often blamed for the fact that, oh, there's so many of them and we don't have any wolves anymore in this area. It's not keeping the deer population down. And deer are important as amplification hosts for ticks that are feeding on the deer, and the ticks have the Lyme disease bacterium in them. And if the ticks get no blood meals, then clearly they wouldn't be problematic. Now, I'm not showing you pictures of rodents, where rodents are actually the key species, many people believe, for amplification of Lyme disease. But the point is that in an ecosystem, if you remove a predator, things can become sometimes more problematic than you'd think. So now I'll get obscure with you and talk about the world's oceans, and it's a reminder that there are these very plentiful bacteria in the oceans called cyanobacteria, and they are photosynthetic. So they capture sunlight, they use that for energy, and they grow on it. They are very ancient. You can find fossils of these from some 3.5 billion years ago, which is not that long after life was predicted to, or inferred to have arisen on the planet. And they did us a big favor. In the process of doing this abundant photosynthesis, they changed the atmosphere and they elevated the oxygen, making it possible for large-bodied organisms like you to be on the planet. So you can thank them for that. Cyanophages are their main predator in the oceans, and they outnumber cyanobacteria. And in a way, they must be regulating the numbers of cyanobacteria in the oceans. So where am I going with this? If the oceans, which are providing nearly half of the photosynthesis on the planet and therefore contributing so much to Earth's oxygen levels, if these bacteria were capturing sunlight and blooming, I'm not showing you a picture of that, but these bacteria can bloom to giant sizes, meaning high densities that you can visualize it from, you can see it from space, okay? So they can go through these boom and bust cycles, but essentially the phages keep them more or less constant. So that, in a way, is keeping the oxygen level constant on Earth, which is a good thing, okay? If it's going through boom and busts and increases and decreases wildly in oxygen levels, then a lot of the species on the planet would be in big trouble. So I now want to go to one thing that was seen in sequencing of cyanophages. They carry photosynthesis genes, okay? And that's a strange thing. What it means, I'll just go to the punchline. If you have a phage and it's infecting a bacterium, it's got to keep the bacterium's molecular machinery churning in order to make baby phages. And it was mysterious to see, well, why do these phages have photosynthesis genes? That's because they bring to the cell the ability to keep the cell churning and keep energy going as they're in the process of killing the cell and making more progeny. So what happens is that the oxygen in the atmosphere can sometimes be coming from these genes in cyanophages instead of genes that are residing elsewhere that are responsible for photosynthesis. So how many of your breaths today do you think you must thank the cyanophages in the oceans for producing the oxygen that you breathed in? Was it 1 in 20 of your breaths, 1 in 100, 1 in 1,000, none of them at all, or every single breath that you took? Not so easy, this one, is it? This is not Australia. What do you think? All of them? I'm hearing all. A. Okay? That's pretty impressive to me, especially as you sit here and you listen to me gab for an hour. If you count up how many breaths you take, a lot of them came from cyanophage genes creating the oxygen from their photosynthesis genes. Okay, so this one is probably the trickiest one. I'm going to go immediately to the next question. So at some point in time, somebody did this crazy thing. They traded something related to viruses for 4 tons of wheat, 8 tons of rye, 4 large oxen, 8 large pigs, 12 large sheep, 2 hogshead wine, whatever a hogshead is, 4 barrels of beer, 2 barrels of butter, 1,000 pounds of cheese, 1,000 pounds of cheese, 1 bed, inexplicably 1 full dress suit, and a silver goblet. So, somebody traded one thing for all of that, and it related to a virus. Was it a vaccine, a flower, a ship, a horse, or a royal family member? Yeah, you thought they were all going to be Australia, weren't you? All right. Any idea? A, E, C. Who here is a horticulturist? You should be ashamed you didn't get this right. So, during the tulip craze, all of that stuff was traded for a single bulb of this kind of tulip, a broken tulip that had this amazing color pattern, and it didn't have only one color, it had 2. That's because it was infected by this virus, and the plant was producing these bulbs that make broken flower coloration. Okay, so that you can easily do through genetic engineering now. You can just make these tulips that have the broken color formation. But anyway, so now we're going to end with whether viruses can help solve health problems. As we heard at the introduction, antibiotics have worked historically, but not very well lately. So all the time you hear in the news of MRSA, which is a form of an antibiotic-resistant Staph aureus bacterium. You can hear about extreme drug-resistant tuberculosis. So there's all sorts of very nasty illnesses caused by bacteria that are now resurging because we are running out of antibiotics to treat them. So this is a very sobering picture. I'm going to get to better news in a moment, but by 2050, especially in the most populated parts of the world, this is annual deaths projected for drug-resistant infections, okay, to the tune of maybe 4 million people a year will be dying in Asia. And also in North America, right at our doorstep, hundreds of thousands of people will be dying. So this is what people talk about when it may be the end of the antibiotic era. So what you can do about it is look to an old idea and try to update it. So phages, I've talked about them, they kill only bacteria, and they could be efficiently used as drugs that make copies of themselves. So now pretend that this growth cycle where you're seeing the phage come in and infects the cell, it makes progeny viruses. So if this is a pathogen, this is different than an antibiotic because it's not only killing the cell, but it's making copies of itself to go kill other cells of similar type. So you shouldn't have to take as high of a dose because the thing creates its own increased dosing. So this was adopted long ago by the Russians, Polish scientists, and now former USSR country, Georgia. So when Western countries were investing in antibiotics, other countries were not. It's far cheaper to do this kind of technology to the point that a lot of Russian soldiers were given vials of phage when they were out in the field in case they got wounded and they couldn't get to medical care fast enough. It's a good way of rubbing that on the wound in order to protect your wound from having an invasive bacterial infection. Cholera is a bacterial disease. It's another one for which rehydration therapy should work and get you through it. If you give somebody cholera bacteria specific phages, in other words, viruses that kill cholera bacteria, they recover faster. That's been shown. So there's an obvious problem, though. We live in a world full of bacteria that have evolved to be resistant to antibiotics. So if you go around using phages instead, I would predict as an evolutionary biologist, you're just going to select for phage-resistant bacteria instead. So this is the question we've grappled with for a couple of years, is whether you can develop a strategy that gets back to this old idea and updates it, because I know that this is inevitable. I know that the phage will be seen as a deadly predator, and the bacteria are going to evolutionarily respond to be resistant to them. So the way this works, the way that we think we've solved that problem, is that in my career I've studied evolutionary trade-offs. Evolution often involves compromises. So something is seen as a challenge, organisms adapt to better meet the challenge, but it doesn't mean that they're becoming super fit in all ways. They might be improving in fitness in one thing that's important for selection, and they might actually be doing worse at something that they had done better in the past. So selection can improve a trait at the expense of another. Humans walk upright all of the time, and other great apes do not. I don't hear gorillas and chimps complaining about neck pain and lower back pain. Well, if they could talk, maybe they would, but you don't expect them to because they shouldn't have it. Their musculature and their architecture shouldn't create those problems, whereas that is a problem in humans. Another example would be these two organisms are quite familiar, they're very successful in their environments, but they're built differently. Cheetahs are adapted for speed and they can chase down a gazelle. If they tried to chase down a zebra, jump on its back, they'd probably break their fragile, long bones in the process. Whereas a lion is not fast enough to chase down a gazelle, but it is big enough and fast enough to chase down a zebra, jump on its back, break the zebra's back, and get a meal. So they're very successful in their environments, but they've been taken to an adaptive solution that doesn't make them good at exploiting the entire environment. So back to phages and bacteria. Our innovation is we look for phages in the natural world, out in the environment, that have evolved to do something interesting, and that is to attack bacteria by binding to what are called virulence factors of pathogenic bacteria. So that's illustrated in this cartoon. Imagine this bacterium, and it's got some proteins on it in yellow and blue, decorating it for a variety of purposes. But in this purpose, these are important proteins that contribute to the virulence of this pathogen and causing disease in humans. So you can go find phages that are specific to these different proteins. And if you can see at the bottom here, it sort of pushes the bacteria evolutionarily to a solution where they avoid phage attack, but they lose or they alter those binding spots, making them less virulent. So you've got a drug that works, and when it fails, it works, because you've still pushed the system to a lower pathogenicity. Okay, so I hope you understood that. I went through that kind of quickly. So there are lots of things that decorate bacteria that are virulence factors. And if you can make it out in this cartoon, I'm showing this red-colored phage interacting with a pilus that's coming out of this bacterium that's important for something like burrowing into the intestine. Okay? There might be things that allow the cell to bring nutrients in and out that might, or even iron, something like that, iron uptake to make it good at scavenging iron from your body and contributing to its pathogenicity. And that could also be a binding site for a different kind of phage. So you just work your way around this bacterium, and you can think of many things that are already known as virulence factors that might attract phages to infect them. So this is one we keyed in on recently. It's called an efflux pump. It's responsible, I think I have another picture of it. It's responsible for, if antibiotics get into this cell, they're actively pumped out. And it can make some bacteria kind of pan-resistant to antibiotics. So we've been looking for phages that interact with these protein structures and try and force the bacteria into a corner. They're either going to evolve to escape the phage, or they'll die from phage attack. And when they evolve to escape the phage, we're finding that they're less effective at pumping antibiotics out. They flip to become sensitive to antibiotics rather than resistant. So that's shown in this diagram. Again, efflux pumps are decorating that cell. An example would be, I'll show you in a moment, Pseudomonas aeruginosa. It's quite a famous pathogen that is loaded up with efflux pumps. So this cut through of the bacterial cell with these proteins showing the antibiotics going from the inside of the cell to being pumped out, that's the same protein that this phage is interacting with. And it pushes the bacteria into that solution. They'll evolve to escape the phage, but instead of maintaining resistance to antibiotics, they suddenly become sensitive to antibiotics. And the physician is going to have a lot of options in using an antibiotic to kill these bacteria and sort of reverse the problem that we're seeing around the world. So I wouldn't be talking about it unless it worked, right? So we did find a phage. The first one we found was in 2016. We were looking at aquatic samples in a lake in Connecticut called Dodge Pond. Pretty randomly, science is full of serendipity. We found a phage that did exactly what I showed you in that cartoon. It forces the bacteria to trade phage resistance for antibiotic sensitivity. And it turned out that we quickly found of a man who had run out of therapeutic options at the hospital in New Haven. He had an artificial part of his heart, an aortic arch replacement surgery, that became infected with what's called a biofilm, or a very tight chronic infection of bacteria that they had no drugs to remove the bacteria because they were pan-drug resistant. So we talked about in many places in the media, and people wrote about this man. He volunteered for the surgery. Sorry, it's not surgery. Actually, he was too elderly to undergo this surgery again. So instead, we administered the phage directly to the site of the infection. And the story goes like this. Ben Chan, who's in my group, they're in the operating room with the surgeon and this guy. They're ready to take a needle and try and place the phage close to the site of the infection, and they found too much scar tissue. They couldn't get to it. So instead, they took advantage of what this man unfortunately had. It's called an oozing fistula. His infection was so bad, it had broken through his chest wall and was oozing out. So they took this and they injected it instead there. And it traveled to the site of his infection, cleared the infection, whereas he had this chronic infection for 4 years that his physicians couldn't do anything with, and he went back to work in New Haven as an ophthalmologist. So this is where we are thanking him in his office, and we're giving him a phage plush toy. It's like, thank you for your volunteer surgery treatment that saved your life, and here's a plush toy. So we're doing a lot of what Ben and my lab likes to call pond-to-bedside personalized medicine. So this is an example. This is a picture of Dodge Pond, and it's just one of the many locations we've sampled. And we find new phages to science that have never been known before. And here's a young woman in Texas who has cystic fibrosis, and she had a bacterial infection in her lungs that was, again, pan-drug resistant. So he is handing her this example of the phages that are going to be put in her inhaler, her nebulizer. And she's going to inhale these over the course of the next 8 days, and it saved her from undergoing a lung transplant. And it didn't eradicate the bacteria entirely. That's very hard to do in the CF lung because it's filled with mucus. It's a very good place for bacteria to reside. But she no longer needs a lung transplant, and she's as healthy as a CF patient could be now. And we've been tracking her. So we are also doing this to target MRSA infections and other bacterial infections of the lung, and we've treated very many people, and this is really taking off quickly. We'd like to do clinical trials about this. So literally, Ben every day gets emails about this from somebody in the USA or around the world who has an untreatable bacterial infection, and we're trying to help them with this. And others are doing it as well. There's sort of this resurgence of phage therapy right now. All right. So the question is, why would the FDA think that that's a good idea? Turns out that now it must be about 12 years ago, the US FDA approved that phages could be sprayed on food. And I bet you didn't know it, did you? Yeah? So you can go to the store and purchase food that has been sprayed with phages to protect that food from being colonized by a form of deadly foodborne bacterium. Is it lettuce, apples, strawberries, deli meat, or raw chicken? Who said? D, I'm hearing. A. Deli meat. Okay? So next time you go to Stop and Shop and you get the sliced ham, this is so safe in the eyes of the FDA that you don't even have to see this on the label. It's just sold to you in the store. So that helps people like me who are trying to create these novel treatments because already it's considered safe for human consumption, okay? All right, so I'll end with, that was some upbeat news about how a virus could be good for you. And now I'll end with, you might not even be in the room if it weren't for viruses. So one factoid from the Human Genome Project that surprised people is that you get the genome map for humans, and almost 10% of it is recognizable as viral. That means that this, long ago in the past, what gave rise to humans interacted with viruses and sometimes took up that genetic material and incorporated it into human chromosomes, okay? Some of it, well, it's generally called endogenous retroviruses. That's the majority of them. And some of them can be maybe bad for you if they're reawakened. This is an ongoing area of research. Don't leave the room thinking that this is well-founded and ironclad. It's not. But some people think it could relate to some cancers, some autoimmune diseases like MS, maybe diabetes. There's lots of ideas out there, but they're very much under ongoing research. Something that is known is that useful proteins, so genes that make useful proteins can come from these genes that are recognizably viral and they're in your DNA, especially syncytin protein, which is essentially essential for placenta formation. So these come from viruses that long ago were infecting things that gave rise to humans one day, actually to all placental mammals at one point. And these are viruses that are quite good at hiding out from the human body and avoiding detection so that they're not fought, which in a way you can think of as a baby developing in mom might be perceived as something that is a foreign body that should be rejected. And instead these proteins from these genes kick in to say that no, that's not the case. This organism should be there, okay? So, thank you. Thank the viruses, because